Hello friends, welcome to my channel Purpose of Learning. Today we are going to discuss one of the topic of pharmacology, that is beta lactam antibiotics. The purpose of this presentation is to know what are antibiotics and what are beta lactam antibiotics, how they are killing the bacteria, its mechanism of action, and which bacteria are most susceptible towards beta lactam antibiotics, and why those bacteria are only susceptible, why not others, and how these bacteria getting resistance to antibiotics, what are beta lactamase inhibitors, and also finally we can also discuss what are beta lactamase inhibitors. This is our agenda, today's agenda. Let's dive into the topic. Beta lactam antibiotics. Beta lactam antibiotics are the major class of antibiotics which have beta lactam ring in common. These are the antibiotics. Beta lactam antibiotics are the antibiotics which are major class of antibiotics. These are the major class of antibiotics and which have one common thing, that is beta lactam ring. In all beta lactam antibiotics, drugs under this beta lactam classification are will contains the beta lactam ring. These are the major class of agents for treating bacterial infections. Beta lactam antibiotics specifies the spectrum of activity from extremely narrow spectrum to extended spectrum. Some antibiotics are narrow spectrum and some are extended spectrum. For example, beta lactam is sensitive. Those antibiotics which are sensitive to beta lactamase enzyme are comes under narrow spectrum actum. Narrow spectrum means these antibiotics will act on small number of bacteria and small number of types of bacteria, and those bacteria which are beta lactamase resistant penicillins and also cephalosporins which are extended spectrum means they act and kill or attenuate the growth of several type of bacteria. These are all the beta lactam antibiotics. One of the advantage here is all these antibiotics possess single mechanism of action, that is we are going to discuss now. Mainly four classes of antibiotics, beta lactam antibiotics are there. First of all, penicillins, cephalosporins, carbapenems, and monobactams. These are all the penicillins, and we can see here cephalosporins. There are five generations of cephalosporins we are included here, and then carbapenems and monobactams. These are all the beta lactam antibiotics, and then discuss general mechanism of action. Bacterial cell wall is made up of peptidoglycan layers. As we know, gram-positive bacteria contains cell wall which is made up of 50 to 60 layers of peptidoglycan layer. 50 to 60 layers of peptidoglycan. Here we can see here in name which contains peptidoglycan layer contains one peptide chain and glycan chain which is formed by peptide chains and glycan chains. If go some more deep, the glycan chain is composed of repeating units of NAM and NAG. This glycan chain, this glycan chain, peptidoglycan chains contains peptide and glycan chain. This glycan chain is made up of NAM unit and NAG unit. NAG means N-acetyl muramic acid and NAG means N-acetyl glucosamine. And then come to peptide chain. Peptide chain contains 5 amino acid chain. This peptide chain is made up of 5 amino acids. These 5 amino acids are always linked to this NAM unit. Then cross-link these 5 amino acids of NAM is cross-linked with another NAM unit of another layer having 5 amino acids, and these are bonded by forming bridge through pentaglycine bridge. The bond between 5 amino acids of one chain, I will explain here. It is clear to you all. Here we have one NAG unit, and this is the NAM unit. This is called glycon chain, and this is the peptide chain. Here we can see here that there are 5 amino acids, 5 amino acids of peptide. Here we can see pentaglycine bridge. Pentaglycine bridge. Actually, this is the cytoplasm, and this is the plasma membrane of cell, and these are the peptidoglycan layers of gram positive bacteria or gram negative bacteria, and these are one. This is one peptidoglycan layer, and this is another peptidoglycan layer. Here we can see the NAG, NAM, and this is the peptide chain amino acids. This is the pentaglycine bridge, which is formed between another NAG and NAM unit of another peptidoglycan. And the formation of this pentaglycine bridge between two peptide chains is known as transpeptidation. This process is known as transpeptidation process. This is the important thing we have to remember. We can see here transpeptidation is essential for providing stability and rigidity to bacterial cell wall. To maintain rigidity and strength of cell wall, this pentaglycine bridge is essential for maintaining rigidity and stability of cell wall. So here one layer of pentaglycine peptidoglycan layer is formed. This is how it is formed. Then now come to our beta lactam antibiotics acts. Mainly this transpeptidation this transpeptidation process is done under penicillin binding proteins. Through these penicillin binding proteins, this transpeptidation process will occur. But what our antibiotics like penicillin, cephalosporins, carbapenem, monobactams were doing here is they are they are looks similar, quite similar to this NAGNAM and peptide unit. They looks similar to this peptidoglycan. So this our penicillin is binds to these penicillin binding proteins. These penicillin binding proteins. The penicillins will binding to penicillin binding proteins. What will happen if penicillins will bind to this? There is no availability of penicillin binding proteins for the transpeptidation process. So the process of transpeptidation will be inhibited. So the inhibition of transpeptidation will leads to the malformation of peptidoglycan layers. There is no formation of this pentaglycine bridge between two peptides. So this will leads to malformation of peptidoglycan layers. This will leads to breakage of cell wall of bacteria and Entrapment of outer solutes into the cell may leads to cell death of bacteria. This is how antibiotics will act on bacteria. This is a biosynthesis and cross-linkage of peptidoglycans is carried out by penicillin binding proteins. Through these penicillin binding proteins, this transpeptidation process will occur. So, penicillin inhibits this transpeptidation process by covalently binding to penicillin binding proteins and inactivates them, result in imperfect bacterial cell wall is formed, which is weak and weak in nature and susceptible to lysis by solutes in the surrounding medium. In addition to the above action, these penicillins also causes activation of autolyzing enzymes. This penicillins also activates autolyzing enzyme, that is murine hydroxylase. What this murine hydroxylase enzyme will do means which initiates cell lysis and causes death. This murine hydroxylase enzyme will initiate cell lysis and cell lysis means breakdown of cell. So the cell will death. Therefore, penicillins are the bactericidal drugs that not only inhibit cell wall synthesis but also causes bacterial lysis. So the penicillins acts by two mechanisms. One, it will inhibit cell wall synthesis and also causes bacterial lysis. Since mainly cell wall synthesis occur during growth phase, penicillins are active against rapidly multiplying bacteria. So the cell wall is mainly formed during the growth phase of bacteria. So these penicillins are more active during growth phase of multiplying bacteria. Then comes to antibacterial spectrum. These beta lactam antibiotics are only mainly effective against gram positive bacteria than gram negative bacteria, because in gram positive bacteria there is a up to 50 to 60 layers of peptidoglycans. As we discussed earlier, the bacteria of the cell wall of gram positive bacteria is made up of 50 to 60 layers of peptidoglycans. So our main mechanism is inhibition of transpeptidation. So inhibits the formation of cell wall of gram positive bacteria, which leads to cell death. But in gram negative bacteria, there is only one or two layers of these peptidoglycans, and also okay, we can see here gram negative bacteria is covered by outer membrane which contains the porin channels. These channels allow selective substances only. So in gram negative bacteria, there is only one to two layers of peptidoglycan layer and which contains the outer membrane. Extra which contains outer membrane which is strong and also which contains porin channels. They only allow specific substances into the cell. So the beta lactams cannot enter fully into the cell. So it cannot active more active against gram negative bacteria when compared to gram positive bacteria. So beta lactam antibiotics are active against gram positive bacteria. Most mostly active against gram positive bacteria than gram negative bacteria. Here we can see some examples of gram positive bacteria and gram negative bacteria. These are cocci, bacilli, streptococci. These are the susceptible bacteria for beta lactam antibiotics. Here we can also see some gram negative bacteria, but these these are mainly acts against gram positive bacteria, although it also acts on some gram negative bacteria like gram negative cocci, E. coli, H. influenza, Neisseria gonorrhea, Salmonella, etc. Next, bacterial resistance. Then we will discuss about how this bacteria getting resistance to antibiotics. For example, take an penicillins. Here it was they are getting resistance by four mechanisms. One is degradation of antibiotics. First mechanism is degradation of antibiotics. Let's take an example of penicillin. This penicillin is entered into the cell to inhibit the process of transpeptidation. But due to some enzymes like penicillinase, cephalosporinase, what will happen means these enzymes will break down the penicillin and convert into penicillinic acid. So it is an inactive substance which cannot inhibit the process of transpeptidation. So the bacteria will survive and action of this penicillin will be inhibited. This is one mechanism, and we can see here alteration of antibiotic binding sites. Actually, these penicillins are binding to which site? Penicillin binding proteins, which will binds to penicillin binding proteins and covalently bond to that one, which may leads to unavailability of penicillin binding proteins for transpeptidation process, leads to malformation of that peptidoglycan chain. So what will happen here? This bacteria will change the will change the what we call structure structure of penicillin binding proteins. So our penicillin cannot bind to penicillin binding protein. So it will do its work like causes transpeptidation and forms a strong cell wall. So the bacteria will resist to this penicillins. This is another mechanism, and we can see here third mechanism is alteration or loss of drug entry pores. As we seen in gram negative bacteria, they have special type of porine channels. They can allow only specific type of substances. So by altering this porine channels, the our drug, for example, penicillin cannot enter into the cell of bacteria. So it can also inhibit the transpeptidation process. Here, the fourth mechanism is increased expression of efflux pump. What means even though the drug entered into the cell, due to the efflux pump of bacteria, the drug in the cell will efflux out of the cell. So it cannot act on the cell membrane. These are the four mechanisms through which the bacteria getting resistance to the antibiotics. Yeah, these are all the things we are discussed right now. This is the this is first mechanism, penicillinases and cephalosporins, and this is resistant bacteria through low affinity for penicillins. By causing low affinity towards penicillins by changing its structure, penicillin binding protein structure, by changing the penicillin binding protein structure, which may cause us a very low affinity of penicillin binding and other beta lactam antibiotics. The example of these resistant bacteria are streptococci, enterococci, and methicillin resistant staphylococcus aureus. so on. And gram negative bacteria alter pore channels, as we discussed here before. Some gram negative bacteria efficiently eject the drug from the periplasm before it can bind to penicillin binding proteins. Example of those bacteria are Salmonella typhi. And finally, last topic is beta lactamase inhibitors. Beta lactamase acts by breaking the beta lactam ring, which results in loss of antibacterial property of antibiotic. Examples of these is penicillinases and cephalosporins. As we discussed here, the enzymes penicillinases and cephalosporins, which may break down the penicillins and inhibits the action of beta lactam antibiotics. Same here, these are the beta lactamases. Examples of penicillinases, cephalosporins. The agents which inhibit the action of these penicillinases and cephalosporins are called beta lactamase inhibitors. What we are doing here, we are inhibiting the enzymes which causes the breakdown of penicillins, which are penicillinases and cephalosporinases. After breakdown of these will produce penicillinic acid and cephalosporinic acid. Eh, Here, these are the examples which inhibits the actions of these beta lactamases, which are clavulanic acid and sulbactam. Actually, we will get the drugs mostly for infection with combination products of clavulanic acid with penicillins, cephalosporins, example like that. They will give along with clavulanic acid because this clavulanic acid will inhibit the beta lactamases. Then the actions of penicillins or any beta lactam antibiotics will be increased. Yeah, here we can see the structure of different beta lactam antibiotics, and this is the beta lactamases which may break down the beta lactam ring of cephalosporins. This is a monobactam, and here this beta lactam will break down the penicillin beta lactam ring. So our clavulanic acid and sulbactam, tazobactam, what will they do means these drugs binds to the catalytic site of beta lactamases produced by gram positive as well as gram negative bacteria, prevent the hydrolysis of penicillins. These clavulanic acid will bind to these beta lactamases and inactivate them, so the hydrolysis of penicillins was prevented. So these are the different beta lactam antibiotics: carbapenems, carbazepines, monobactam, cephalosporin. We can observe here every beta lactam antibiotic contains the beta lactam ring. Yeah, is it clear? I think, hope you all understood the mechanism of action of beta lactam antibiotics and antibacterial spectrum, to which bacteria these antibiotics are more susceptible, and what are the bacterial resistance mechanisms, four mechanisms, and and finally beta lactamase inhibitors: clavulanic acid and sulbactam, tazobactam. Thank you. Thank you. Bye bye.